In this video, I'm going to present my research on medical devices and biorobotic systems development. I will start with my current research activities and then provide a quick overview of my future research plans. Before I develop more on my research, let me give you an overview of my current affiliation. I'm currently an assistant professor with the University of Ontario Institute of Technology, now known as Ontario Tech University, located in Oshawa in the suburbs of Toronto. I've been assigned to the Mechatronics program. Within the Faculty of Engineering, we have around 4,000 students, and we have 8 programs including Mechatronics, and that's where I work. I have been with Ontario Tech since July 2017, and in January 2018, I established a research lab that you see here at Ontario Tech called the Biomechatronics Lab. My research focuses on applications of mechatronics to biomedical engineering, including medical device development and imaging methods for robotic surgery. My main areas of interest are summarized here. The first one is robotic surgery, the second is haptics, and the third one is instrumentation. At the intersection of robotic surgery and haptics, we have teleoperation, and my research projects related to teleoperation include telesurgery, collaborative human-robot surgery, and the development of precise medical devices for robotic surgery. At the intersection of haptics and instrumentation, we have force feedback. Research here focuses on two things: on creating force feedback by developing force feedback devices such as haptic devices, and also how to develop instruments that can measure the mechanical and electrical properties of tissues and use that information to create force feedback. At the intersection of instrumentation and robotic surgery, we see image-guided surgery. In this sub-area, my research focuses on developing surgical tools or robots, diagnosis tools such as systems for real-time biopsy, and new imaging methods for robotic surgery. All these three areas are complementary, and my lab lies at the intersection of these three fields. In the upcoming slides, I'm going to give some examples of robotic surgery, instrumentation, and haptics, and how we combine them in the lab. To do that, I've chosen these topics to present today. The first one is robotic-assisted surgery, and to illustrate that, I'm going to use the example of percutaneous nephrolithotomy. The second topic I would like to present today is robotic-assisted brachytherapy. Brachytherapy is a needle-based procedure that benefits from the assistance of robots to achieve more precise targeting in tissue. The third topic is instrumented needles for tissue discrimination. In this part of the presentation, we're going to see how we can instrument needles to measure the electrical properties of tissue and then use them for either tissue classification or to implement force feedback during robotic surgery. We are then going to move on to instrumented needles for tissue imaging. So instead of using a single needle for biopsy or local tissue discrimination, the idea is to use several needles at the same time, all having the same sensors, and then use the communication between them to create images instead of just localize the tissue at a given point. Finally, I'm going to conclude with a short and long-term research vision. Percutaneous nephrolithotomy, or PCNL, is a procedure recommended to remove kidney stones when the patient has kidney stones that are blocking certain branches of the kidney or those stones that are larger than 2 centimeters in diameter. This procedure involves making a small incision in the patient's back and insert a small instrument towards the kidney, navigate that instrument in the kidney, and then reach the stone. When the stone is reached, they are either removed or they are first broken down into smaller pieces using ultrasound before being removed through that tube. Despite the fact that this procedure has been around for 40 years, it is still a very complicated procedure for surgeons to learn and perform. In fact, in the USA, around 90% of the surgeons are unable to gain proper kidney access to remove the stones efficiently. Marion Surgical is a company based in Toronto that has developed a surgical simulator to help the surgeons train and develop their skills and also simulate different operating scenarios. We partnered in 2018 through an NSERC Alliance grant, and the objective of our collaboration is twofold. First, they have issues regarding the haptic feedback that they have in their console. The problem with their simulator is that their device that was built in-house is not very transparent. You can feel the inertia and friction of the device, which compromises the fidelity of the forces that would come from the simulation of tool-tissue interaction. The first objective of our collaboration was to develop a new system to improve the haptic feedback there. The second objective of our collaboration is to use the simulator to move towards robotic assistance. So the idea is to use the simulator to simulate different scenarios, acquire the data, and based on that data, create some sort of optimal tool path for different operating conditions. Once that a tool path is defined, we can now bring in a robot to help the surgeon manipulate the tool and reach the kidney stones with more accuracy. And this is what we developed here. This is the simulation of a kidney in the lab, a kidney model, and here we have a 6-stuff robot arm from Academic and a haptic device. The idea is to use the simulator to create the optimal path that will guide this tool from the entry point in the kidney towards one of these stones. By controlling the position of this 3DOF device, the position of the tooltip can be controlled, and the robot is programmed in such a way that it maintains always the same entry point in the tissue. This way, we can convert a 6DOF problem into a 3DOF problem. When it comes to learning from demonstrations, it works as follows. We use the simulators to develop these trajectories that are the optimal trajectories between the entry point in the tissue towards the stones. We convert these points into an optimal trajectory and create a potential field around them. This works like a valley that we see here, and at the bottom of this valley we see these trajectories. The magnitude of this graph now represents the magnitude of a force that this haptic device will apply to the hand of the operator as the operator deviates from that trajectory. You see, the robot is not doing the operation by itself, it's supervised by the human operator here. The human operator controls the position of the haptic device, and if the position deviates from these optimal trajectories, then we see this force has been applied. You see the vector field here, moving the hand back towards that optimal path. And this is the same representation in 3D. There are a few challenges here to implement this, besides the haptic feedback. The first one is that the robot that operates on the kidney is a 6DOF device, whereas the console we use to control it only has 3DOF. So it needs to develop a constraint kinematics that makes sure that this entry point in the tissue is always fixed. The second issue that we have here is that these joints are prone to saturation. To solve this issue, we implemented a numerical inverse kinematics. We take the position of the haptic device that will correspond to the position of the tooltip. We also take the position of the entry point in the kidney, and we make sure that the tool always passes through that point. So the inverse kinematics must make sure that the tooltip goes to the position of the haptic device, and this specific point along the shaft that will move up and down always passes through that point. In addition, to avoid saturation of the joints, that is, the joints will not reach their maximum limit, which would invalidate the inverse kinematics, we introduced this new saturation function to the position of each joint. We see here the actual position that it would take from the inverse kinematics, the traditional inverse kinematics of a 6DOF device, and you see here this tangent function that will saturate the joint to maximum and minimum limits that we can specify. We can then write the inverse kinematics as a function of the Jacobian of this robot that now takes into account this transformation of variables. And this is an example of the inverse kinematics. We have a desired position of the tooltip that corresponds to the position of the haptic device here. We create a position error and use this error to create the velocity of each joint here. So the entire inverse kinematics takes into account this saturation. Once the kinematics is completed, we convert the variable back into the joint angle that is visible in the robot and then send that to the forward kinematics. Another difference here is that Jacobian is defined strictly in Cartesian space. X is the position of the tooltip and also the position of point number 7 here, the entry point in the tissue, unlike traditional inverse kinematics where you would have position and orientation. This makes things a lot more complicated. Here is one example of one element in this matrix. There are 58 of those. But this ensures that the joint limits are not saturated and the constraint of passing always through that entry point in the tissue is always respected. To validate this approach, we conducted experiments with 3 groups of users. The first group performed the surgery without any haptic feedback. They were given a simple task of going from the entry point to one of the stones. The second group was provided haptic feedback. That is, we defined the optimal trajectory for them, and they would use the robot to go from the entry point in the tissue towards one of the stones without knowing where that optimal trajectory was, but of course receiving haptic feedback to guide them along that trajectory. The second group was trained with the haptics exactly as group number 2, but then they were allowed to perform the operation without any haptic feedback. Here are the results. We assessed their performance in terms of time they took to complete the procedure, path length from entry point to one of the stones, and average deviation from the so-called optimal trajectory. And we see a very interesting trend. The group that performed the operation without any haptic assistance shows a very large standard deviation in all 3 criteria. The second group shows a significant improvement, so this group had haptic feedback throughout the entire procedure. We see that they spent less time to complete the procedure, the path is more direct towards the stones, and the average deviation, without any surprise, is smaller than compared to the case without any haptic feedback. An interesting observation can be seen in the third group. We can see that they perform a little worse than group number 2, but they are better than group 1. This suggests that this system effectively taught them how to perform the surgery. And this is the main conclusion here. The haptic assistance not only makes this group that uses them better than the group that didn't use it, but it also teaches these users how to perform the surgery more efficiently. In all these tests, we are using this 3DOF device. This is not really meant for research, it's more like a gaming console. The performance aren't great, so we decided to build our own. And here is what we built. This is a 3DOF device that can be used for the same purpose. The main difference can be seen in the actuation mode here. Instead of using electric motors, we used rheological brakes. So this system becomes now completely passive. It cannot go unstable, which makes sure that the entire teleoperation scheme is stable, and also will provide a much better transparency compared to the other device we saw. We defined these simulation surfaces and programmed the device to always apply a force when in contact to it. We can see here that as we move the end effector of the device, the end effector doesn't penetrate that, it always moves around it. So even though this is a passive device, we got pretty good performance here when following this sort of surfaces. We're now going to move on to robotic-assisted brachytherapy. Brachytherapy is a type of internal radiation therapy, often used in prostate and breast cancer treatment. The idea for both types of treatment is the same. Long needles are inserted into the tissue where the tumor is. Here is the example for prostate cancer and here for breast cancer. Here are the needles used, and several needles are inserted through this guiding template into the tissue or near the tumor. Once in place, radiation is delivered to the tumor. We have a similar setup for prostate cancer. We see here this brachytherapy template being attached close to the perineum, and the needles are inserted through them. Specifically for prostate cancer, we can also notice here a transrectal ultrasound probe that will take images. There are 2D slices of the prostate here, and this is the only feedback the surgeon would have to guide the needles towards their destination. The procedure works as follows. We take a set of axial ultrasound images that we see here. We now overlay these images on the position of that guiding template. So these are all possible positions where the needles can be inserted. And based on this, based on the contours of the prostate in this case, we can build a dosimetry plan that will specify where needles should be inserted and how much radiation should be given at each of those locations. There are several problems with this approach. The first one is that needles are very hard to localize in the tissue. The second one is that if you look at this image, it's not clear where the tumor is. Because of this, because you don't have a clear image of the tumor, even though the tumor sometimes is localized, ultrasound images don't have enough resolution to allow the surgeon to target them. Another problem is that the needle is a continuum robot, meaning that it will deflect, it will not travel straight. If you go back to the setup here, the assumption is that if the needle is inserted in this location, it will travel straight into the tissue. So when the needle is inserted here, it is assumed that it will go straight, but in fact it deviates from that location and it will end up there. The dosimetry plan that is created may not be accurate, and that's another reason why the entire tissue needs to be treated, even though the cancer may be localized. The solution to this problem is to treat this as a feedback control system. This would require modeling the needle-tissue interaction, estimate future deflection of the needle, and use some sort of feedback control to reach the destination in the tissue. And this is one of the devices that we proposed. This device has a brachytherapy needle attached to it, and inside here we have an actuation unit. The objective, if you look close at the tip, is simply to rotate the needle or allow for these rotations to happen to correct the trajectory. The needle has a bevel tip, and if inserted in the tissue, it will just deflect away from a straight line. But because of the bevel, if you now rotate the base of the needle, it will actually deflect in the opposite direction. So we can use this, say, limitation to our advantage. And if now we are able to control the needle rotation precisely based on a needle-tissue interaction model, we can make sure that it always reaches the target. So inside of this device, we see a actuation unit that can rotate the needle. And we also have here a piezoelectric actuator that vibrates the needle at a high frequency so that needle-tissue interaction due to friction along the shaft is minimized. We can treat now this as a feedback control system. The first one is the device itself that we just went through. The second is the tissue and the needle interaction model. In this case here, simply model as a cantilever beam that reaches a certain equilibrium, and you can also model the tissue compression above and below the needle. The next part is the sensor. We need some sort of feedback, and in this case here we could simply use ultrasound images to make sure that we can track the location of the needle at all times in the tissue. This gives the actual location of the tip that you can now use for trajectory control. The next stage is trajectory planner. So if you take this needle tissue interaction model and you know how the needle reacts to insertions and rotations, we can now create an optimal trajectory from an entry point towards a given target. Based on that, we can now explore many different possibilities of rotation and insertion and create a plan that corrects for the deflection of the needle as it goes. This leads me to the third part of my presentation, instrumented needles for tissue discrimination. This is motivated by one of the issues we discussed in robot-assisted brachytherapy, the fact that it's very hard to locate the tumor from ultrasound images. What if we take these needles that are already used in the procedure and instrument them to deliver information about the tissue at the tip, and perhaps use that information to classify the tissue as these needles are inserted? This is what we studied here. The objective of this project was to instrument the brachytherapy needles for real-time tissue classification, and later for use in haptic feedback. This is one of the designs of the instrumented needles we developed. Here we simply have the needle shaft. They have a copper wire fed through the shaft. We have one electrode at the needle shaft itself, and the second electrode is the copper wire at the needle tip. The second configuration is seen here. The needle shaft acts as one of the electrodes, and that is stylet, as an insulating material, and only the tip is exposed. So here we have one of the electrodes, here we have the other one, and this arrangement allows the stylet to be removed from the needle, freeing the shaft so that drugs or radiation can be delivered. This needle is connected to this spectroscopy circuit. So the needle here is the tissue. We have one of the electrodes here, one of the electrodes there. This is a very simple arrangement that was custom made by a company for us. This system delivers a current from the primary to the secondary electrode, and we also measure the voltage difference that creates. So we see that this part here is simply a current source, and this second part here is a differential voltage measurement system. We can do this at 23 frequencies that will range from 10 Hz to 250 kHz. By analyzing the magnitude of the current delivered at a given frequency and the measured voltage, we can calculate the tissue impedance. And to do that at 23 frequencies, we now have a spectrum, a signature of the tissue that we can use for tissue classification and perhaps even real-time biopsy. We tested this in many different tissues. Some of the samples are here. These are ex vivo tissue from grocery stores. These are sample tissues with gelatin and porcine phantoms. And we also went to a local vet clinic where we could have fresh tissue, and we measured the tissue within 5 or 10 minutes after the tissue was removed. Here are all the tissues we tested, and you can clearly see that the spectral data that we get from them are distinct. We can now use this to try to classify the tissue. We see the discrete points on this graph are the actual measurements that you get from different tissues. How do we go now from this discrete number of points to these nice curves that interpolate them? To do that, we need to fit this data to a model. And to that end, we use the double dispersion model seen here. So the tissue is treated as an equivalent electric circuit. We have 3 resistors following this arrangement, and you have 2 constant phase capacitors here. We can now create an equivalent impedance model and then try to find the values of all these components that would replicate the spectrum that we see in the measurements. We use 2 approaches. The first one is a single objective, and the second one is a multi-objective approach. The first method is the single objective, and for that we use the modified Newton-Raphson. So the idea is very simple. We have the measured impedance and the estimated impedance, and we're simply creating an error function. This error function is the difference between them, the least square error, over all 23 frequencies for both phase and magnitude combined. We now assign an initial guess for that vector p for all parameters in that circuit, and we are going to iteratively adjust that value by a certain amount delta p. And this is now done by finding the derivative of the impedance model that we created for that equivalent circuit with respect to each element in the circuit. Using this Jacobian and the modified Newton-Raphson algorithm, we can make this converge in a way that this error tends to 0. This is a deterministic method, which means that you only get one answer for this optimization problem, and this answer will depend on the initial conditions. The second idea that we had was to use a stochastic method instead. And instead of treating this as a single objective, go for a multi-objective where phase and magnitude are separate. And this is what we have here. We have a multi-objective, multi-parametric system that will now try to minimize the error between the phase and the magnitude separately. This was accomplished using the non-dominated sort of generic algorithm. The advantage here is that you get multiple solutions. And in this case, we ended up with 25 solutions for each tissue. That's the population we chose. We now take a tissue type, split that into different tissue samples, send that to the optimizer, define the population numbers, create this Pareto front, and now we have a set of potential solutions that will fit the spectral data. We can do this for both training and the data set, and by having the option of going through many possible solutions, we not only eliminate the need for a good estimate of the initial guess as in the modified Newton-Raphson, but we also eliminate outliers from the solution. We have also worked on classification methods that I'm going to skip here because we have tried many different ones, but the idea is to simply use these parameters to now look at the database and classify the tissue based on the database. In addition to tissue biopsy, of course, and classification, there are two applications that we explored. The first one was to tactile feedback. The idea was very simple. We take now the needle that we instrumented, that is connected to the device here, and we attach this vibration motor at the needle shaft. The user holds the needle and inserts the needle in one of the tissue samples. At the tip, we have the sensors. The sensors are measuring the spectral data in real time using the spectroscopy device. We take the impedance data and we pass that through the classifier. The classifier then identifies what tissue the needle tip is going through in real time. We can then assign each tissue to one vibration pattern. Here we have 3. We show this is time and you have the intensity of the vibration. So this is like a ramping up vibration. These are small pulses, and this is one going up and down and back up. So depending on the tissue that the needle tip is going through, the user will fill these patterns and will help them get real-time feedback of what tissue the needle is traversing at the tip. I have 3 different tissues, and you see the success rate. So this is the tissue at the tip, and this is what the user perceived as being the tissue based on these patterns. And you see that the success rate is relatively good here. Overall, 88% of the time, they were able to classify the tissue based on the vibration patterns they received. The second application is to use these needles for haptic feedback. Here what we did was to, instead of using mechanical force, to actually use the electrical impedance of the tissue and then relate that to a force that is displayed to the user. So here the user is controlling this robot using the same haptic feedback we had before, and the forces the user feels here are proportional now to the impedance, the electrical impedance of the tissue. And you see 2 different layers of gelatin. The objective of this experiment was to see if the users were able to, based only on the force perceived without actually seeing the tissue, if they were able to locate where the second layer was. I'd like to draw your attention to these 3 graphs. The first one is the force that we measured at the needle base. This is what the user actually feels if we would just use a conventional approach of measuring the force and mirroring that at the haptic device. We see that this is the gelatin layer, the red layer here, that the force increases because of friction, and when the user punctures the red layer, we see this small spike and then immediately goes down and back up. This is now the impedance that we see, the electrical impedance measure at the tissue. We see that this transition is much sharper. We can now associate this to the haptic feedback, and this is the haptic feedback that the user received. We performed this test. The first one, the users were requested to just insert the needle at 3 different tissues, try to identify the depth of that layer. And the second one, the user used our teleoperation scheme. You see the robot here, the tissue is behind and hidden from view. So they requested to now move the robot to the position where they thought they were entering the second layer and just leave the robot there. And here we see the results. Hand trials in green and the robot trial in blue. We see that for all phantoms that we tried, the accuracy is much better using the teleoperation scheme we proposed. I'm now going to move on to instrumented needles for tissue imaging. We can try to take this approach of instrumenting needles one step further. If you remember from the procedure presented earlier, the surgeon is not dealing with one single needle at a time, but several needles. So instead of looking at the tissue properties at the needle tip, could we use several needles instead and then look at the tissue properties across them and perhaps use that to create an image of the tissue instead of just getting the information about the tissue at the needle tip? The objective here is quite different. Instead of using single needles, we use several needles. Each needle serves as a single electrode, so it only delivers or current or measures voltages. And you can now use electrical impedance tomography and do that from within the tissue. So here is one example. In electric impedance tomography, the tissue is modeled as this finite mesh of resistors. Our objective is to now apply current and measure voltages somewhere in this mesh, and based on those measurements, infer what the resistance of each of these resistors is. Each of these dots in this example will represent the position of a needle. In this example, we have 3 pairs of needles. This one here is chosen to be the one that delivers a current. We can now apply a low current here and then measure the voltage induced across the different pairs. We can now switch the electrodes that deliver the current and measure the voltage elsewhere. Based on these measurements, we now have to identify what the value of all these resistors is. This is an ill-posed problem because the number of resistors that we have inside will exceed the number of independent voltage measurements that we have. There is a key difference here compared to classic electric impedance tomography. In electric impedance tomography, these electrodes are only placed at the tissue boundary. Because you're using needles as electrodes, they can be placed anywhere within the tissue. To solve this problem, we used a discrete approach where we have the voltage at each of these nodes in the tissue. We have a conductivity matrix that is here given by C-1. And we have a current injection pattern. Of course, the only voltages that we can actually use are those that were measured where the needle is. So everything else cannot be used. We only have this finite number of points. We can now take this finite number of points that are measured, compare that with our estimation given here for that assumed distribution, and define the error between the actual voltage measured and the one that we got in the simulation. This will create the least square error of all voltages there. We can now use the same modified Newton-Raphson that we used before to adaptively change C, the conductivity matrix, until this error is minimized. We applied this to brachytherapy. You can see the experimental setup here. We have the robot that is used to position the needles accurately in the tissue, and each needle is connected to the spectroscopy device. Each needle is a single electrode now. This is the brachytherapy template that I presented in the beginning. So now our resistor mesh has 1300 resistors and they are overlaid on top of this brachytherapy template. This gives us for now a resolution of 2.5 millimeters. So between two entry points here we have two resistors, and you see that at the entry point or the possible location of needles will actually match the node in the resistor mesh, will match this holes that you see in the brachytherapy template. We validated this by putting a circular mass inside the tissue and inserted 8 needles around it, and this is the image we got. It's a fairly good representation of the actual mass inside the tissue. There is a big limitation of this approach. As you can see here, we have much more resistors than actual measurements to solve for. So the resolution of these images are not great. There's the intrinsic limitation of electric impedance tomography. And this is the objective of another line of research, is to explore acousto-electric tomography. We know that electric impedance tomography is ill-posed and it has limited resolution. We can use more needles, but it will always reach that hard limit. So the idea is to exploit the acousto-electric effect. Here, ultrasonic pressure that comes from the transducer that is already used modulates the tissue electrical conductivity locally. This has been known since 1946, but it has only been explored on medical imaging a couple of years ago, but never in brachytherapy. If sigma 0 is the original conductivity of the tissue, and we apply an acoustic pressure to the tissue, the new conductivity sigma p will be a function of the acousto-electric coupling parameter K and the magnitude of the ultrasonic pressure. This is a very simplified model, but we can see from this that now these voltages that we are measuring in the mesh will be proportional to the current, as it was the case for electrical impedance tomography, but now they are also proportional to the acoustic pressure applied to the tissue at a given focal point. For what follows, we are assuming that this acousto-electric coupling parameter is a known constant. The idea here is as follows. We have the same mesh as for electrical impedance tomography. We have one pair of needles here that are used to deliver current, and another pair that is placed here that is measuring a voltage V1. We can now program the ultrasound probe to deliver a focused pressure somewhere in the tissue, and let's assume that that pressure will only affect this resistor here. If that is the case, then we have a new voltage measurement because the mesh has changed locally. We can now look at another location in the tissue, which again will change the voltage measurements, and you can continue this process. As you can see, as we steer the ultrasound probe across the tissue, this voltage will change. We can have a lot more measurements as compared to the traditional electrical impedance tomography. In electrical impedance tomography here, we would have actually two measurements, one for this configuration and another measurement when we swap the current and voltage electrodes. In this configuration, we have the same amount of electrodes, but you can have many more measurements that will help solve that inverse problem. Of course, here in theory, this would be enough to solve for the inverse problem. If you're far away from the electrode, this modulation here may not provide significant change in voltage readings. We can add another electrode and now measure voltages there. We can now create a way that if we are applying ultrasonic pressure close to V1, we only trust measurements from V1, and if you're down here, we only look at measurements from V2, and if you are at the vicinity of them, we can then look at both concurrently. Here are some simulation results. We have the mesh that we with 1300 elements from before. We have an injection electrode here. We have a resistance R for this part and 1.6 R inside. This is the mass. We now define an ultrasonic zone here where the pressure is applied. In this case is 1 megapascal. And in this case, we have a smaller zone, but now the acoustic pressure is up to 3 megapascal. It must be noticed that these two pressures here are lower than clinical standards. We can now compare the voltage measurements across the entire zone here with and without ultrasonic pressure. So basically we take the original problem, see what the voltage potential is, we apply the ultrasonic pressure, we see what the potential is, and then we subtract them. And this is what we see in these two graphs. On average, we get around 80 microvolts per amp per ohm. For biological tissue, our estimation is that this will create an average across the mesh of 50 microvolts. That's the change in the voltage readings that comes from modulating the electrical conductivity using ultrasonic pressure. This is a very small voltage that currently we cannot measure with our equipment. And also our equipment is limited to 350 kilohertz, but the ultrasonic pressure here will go up to the megahertz range. So you see two components in this voltage, the low frequency component that comes from the spectroscopy device and the high frequency component that comes from the ultrasound. Right now we cannot measure the ultrasonic component, but this machine is a Verisonics ultrasound research machine that was acquired through the NSERC RTI, is to be installed in the lab in the upcoming weeks, and we'll be able to now get some experimental results using the machine. So the machine will be used for two purposes. The first one is, of course, to control the ultrasonic pressure using acoustic force radiation impulse. And the second use of this machine is to actually use its receiving channels as electrodes. So the needles will be connected directly to the inputs of this machine, which will give us a much higher accuracy in voltage readings and also a frequency range. In the previous slide, we introduced the idea of using multiple electrodes for different measurements. And the question was, which one should be used, for example, where the ultrasonic pressure is applied here? Or this question can be asked in the opposite way. Well, if we have this electrode here, where should the acoustic pressure be applied in order to maximize the voltage readings seen by this electrode? Same for V2. We can take these voltages and take their partial derivative with respect to each element in the mesh, and then create the sensitivity matrix. And this is what you see here. For this input current and for V1, we see the regions where the ultrasonic pressure can provide a significant change in the voltages for V1. So you can create these maps for each individual electrode and then optimize their use in a way that we can always get accurate measurements from each of them. As I said, this is research in progress, something that was started recently, and up to now we've not been able to validate this experimentally due to the lack of experimental resources. But thanks to the NSERC RTI grant, we'll be able to validate this experimentally very soon. We have seen many different applications of medical device in mechatronics and robotics for biomedical applications. For the first part, it is clear that by introducing robotic assistance in the operating room, there are several advantages. We can create new treatment options that are currently not feasible. We can enhance documentation and follow-up, save on operating room time and costs, and shorten hospital stays. There are still several challenges related to bringing robots to the operating room, and those are currently and will be the future focus of my research. Same for new instrumentation methods that I'm developing. There is a clear need for new methods to enable real-time tissue biopsy and tool navigation. We can also use new instruments to enable acoustoelectric tomography and acquire images directly from within the tissue, which will create much better images than those that are acquired externally. These imaging methods, especially acoustoelectric tomography, are new, and there's only little research done on these topics. There is a great potential there, and this type of methods can eventually lead to new treatment options that are currently not feasible. One example is for brachytherapy, where now if the lesions in the tissue are clearly visible, we can move away from treating the entire tissue and just use focalized treatment. The last topic of my presentation is my research vision for short and long terms. I believe that research and development must solve real problems and address real needs. I always try to work closely with industry and end users to make sure that we are solving a real problem and we are addressing a clear need. The short-term projects that I will be carrying on for the next 3 to 5 years are pretty much the ones we discussed in this presentation. The acoustoelectric imaging that I just started, instrumentation for stereotactic biopsy, acoustoelectric tissue discrimination for real-time biopsy, and acoustoelectric tissue discrimination for haptic feedback. So most of these are related to instrumentation, but with an application to real-time biopsy and also an application to robotic surgery, especially when it comes to creating methods for haptic feedback during surgery. Once they are completed, I would like to move on to other projects such as ultrasound tomography, multimodal tomography, not only acoustoelectric tomography, but potentially combine that with magnetic resonance imaging. Also develop a research line on implants for health monitoring, that are some sort of implants that can deliver real-time information of the patient condition, and eventually start a new area on magnetic guided microrobots. So this concludes my presentation. Thank you very much for your attention. If you have any questions, we can discuss that by email. Here is my email. Here is my research lab. You can also look at this website for more information on my current research activities. And this is the lab Twitter. Feel free to follow us there. Thank you very much.